Yeah, so what I will try to do today is I will try to show you how we do things at AnaBios and how we do human translational sciences to try to push forward, novel therapies that patients are really in need. So this slide is simply to give you an idea about what I will be talking about. So I will tell you about how we do procurement of normal and diseased donor heart. I will give you an idea about the different models we have when it comes to support drug discovery, give you a simple overview about the cardiac myosin and how you can modulate this myosin, for heart therapy. So we'll talk a little bit about obstructive hypertrophic cardiomyopathy, heart failure with preserved ejection fraction, heart failure with reduced ejection fraction, and most importantly, we will show you some novel data showing the potential of inhibiting the myosin, to treat arrhythmias. And I will close the talk, by the talk home message. So basically, I don't need to repeat again that failures of novel drugs in clinical trials is still very significant. And one important reason for this can be that the artificial cellular models or the animal models being used in preclinical research, do provide data that they do not translate to patient. And hence, the need for a human-based model to try to understand the human biology, try to understand the human disease. And this is what AnaBios is about. We are a company focusing on human translational science. And how we do this, we work with organ procurement organization in the United States, and we do recover human organ. And you can see, they are listed on this slide. So we develop special solutions that allows us to, when we recover, an organ, and when we ship an organ to our lab here in San Diego, to maintain, the highest quality and the highest viability when we recover this organs. And of course, a good testament for the high quality and viability is our ability, to develop cellular and tissue or slice-based model, to do some drug activity profiling, but also our ability to provide cell samples or tissue samples, to run omic analysis and be able, to go after novel targets. And as I said, one of the organ we recover is the entire human heart. So we can recover normal human heart, but also we can recover diseased human hearts. And this slide shows you, the different models that we have developed that allows us, to support cardiovascular programs at the preclinical stage. So you can see, we can intervene at LO candidate selection phase. We have tissue-based models that allows us or allow a discovery program, to go after the effect of a compound on the electrophysiology of the heart, on the contractility of the heart, and also go after, effect of compound on blood pressure and heart rate. And also, as you know, these models can be used during late phases of preclinical development, but also cell-based models are very important to support the discovery programs during target identification and LI phases. So this is where you need the throughput to be able to support, the medicinal chemistry effort at every company and every project. In addition, as I said, we can do this, we have these models when it comes to normal donor heart, but also we can recover diseased donor hearts. So when it comes, for example, to HFpEF, HFrEF hearts, we can recover hearts or donors, hearts of donors affected with atrial fibrillation. We can go on after hypertrophic cardiomyopathy. We can go after MI. So we have all these models, to try to confirm, the potential of drug to be efficacious when you take it to clinical trials. And again, here, when it comes to diseased donor hearts, we have tissue-based models and also we have cell-based models. And, for the next slides, I will show you how we are using the contractility model, with ventricular and atrial myocyte to support drug discovery programs when it comes to cardiovascular therapeutic area. This is an image of the ventricular myocytes that we do isolate from donor hearts. You can see this cells, they have a very specific structure, very specific morphology. they have this clear striations that they constitute, the contractile machinery in this myocyte. And, to be able to use this model to support early drug discovery, we developed the MyoBlazer, which is a novel non-invasive optical method. And you can see, we can record from multiple myocytes at the same time. And we can, in real time, track the effect of drugs that we are testing. And, everything is automated in terms of acquisition and analysis. And this is an example of a compound that decrease contractility. And you can see, this is a time course of recording from 5 cells at the same time. You can see the first 5 minutes are recording at baseline. And then we tested the compound at multiple concentration. And you can see we can determine the no effect concentration of the compound. We can determine the IC50 concentration. And also we can determine the maximum effect happening at which concentration. And of course, at the end of the experiment, we can look or assess the reversibility of an effect that we may see. Again, from this contractility transient, you can see we can analyze the effect of drugs on multiple parameters. And the parameters listed on this slides allows us to check if the compound is affecting the systolic phase of the contraction or the diastolic phase of the contraction. After doing or assessing the effect of drugs on contractility, always we need to get some mechanistic understanding. And this slide show you, that the calcium imaging model we have, and you can see here also we are recording from multiple myocytes. I believe the number in this slide is 26 myocyte. And again, we can record the calcium transient here, and we can answer the question, is the effect of contractility I see with my compound is calcium dependent or calcium independent? And similar to the contractility transient, again, we can analyze the calcium transient. We can look, for the effect of the compound on the peak height of the calcium transient, but also we can look at the decay of effect of compound on the decay of the calcium signal. Again, we heard a lot of mentioning today about of the human IPS myocyte, and again, we heard that the human IPS are not exactly similar to the adult ventricular myocyte or myocytes that you can isolate from the adult human heart. And this is some data for a collaboration we had with the Francis Crick Institute in London. And the paper is in press in cell report method. And you can see, from the data on the left-hand side of each of the two figures, this is the data using our human ventricular myocyte and our human ventricular tissue. And on the right side of each figure, you can see the data that we're generating using human IPS myocyte. And looking at different important signaling pathway in the myocyte, you can see the huge difference between human IPS myocyte and adult ventricular myocyte. So the main message here, be cautious, when you use the human IPS myocyte to decide the fate of a target you are after, or to decide the fate of a compound that you would like to progress and take forward to clinical trials. And we already published similar data with a collaboration with Boehringer Ingelheim in Germany, where we looked at the difference in terms of ion channel expression between the primary ventricular myocyte and the human IPS myocyte. This is another slide, to confirm again, that human IPS models are not similar to the human primary myocyte. And this is a collaboration we have with the FDA. And if you look at the PCA analysis, you can see the data from our myocyte, from our tissue, and data from tissue that the FDA procured from Tamer Labs. You can see the difference. There is no clustering, between the adult human heart data and the human IPS or the IPS engineered human tissue, and this data shows you some data related to important proteins that they play an important role in the contraction. Again, be careful, when you use the human IPS models, and make sure that you use the human models that we have been developing using the human donor hearts. Again, this is a brief slides and high-level slides on zamaiozine. So as you can see, we have a cartoon here of the sarcomere. So this is the contractile unit, if you want, in the myocyte. And basically, this sarcomere is composed of two main proteins. You have the thick myosin and the thin actin. And basically, the myosin is the molecular motor that powers cardiomyocyte contraction. And you can see once the contraction happens, what you see the myosin slides along the actin to contract a cardiomyocyte. And you can see this is a human ventricular myocyte that shows you how this sliding is happening. This is a slide to give you an idea about the mechanochemical cycle and its regulation when it comes to the myosin. And you can see on the slides you have multiple players here. You have the calcium, you have the troponin C, myosin, actin, you have the ADP, and you have the ATP. So basically, when the calcium binds to troponin, you can see the myosin head binds to the actin, and this will lead to the ADP and the inorganic phosphate to be released from the myosin head. And what's happened after this, the myosin heads will pivots, bends, and pulls on the actin and moving it, causing the myocyte to contract. And then, you have the ATP will bind, to the myosin head and causing this head, detach from the actin. And again, you will have the ATP will be hydrolyzed into ADP again, and you have the cycle, happening, and again, you have contraction after contraction happening on the basis of second by second, basically. So when it comes to modulating the myosin for heart therapy, when you modulate the myosin, basically you modulate the heart contractility and you modulate the relaxation of contraction. And I will show you some data how the ventricular myocyte and using compound targeting the myosin, you can use it for hypertrophic cardiomyopathy treatment, HFpEF, HFrEF, and anti-arrhythmic therapy. These slides give you an idea about the drugs that we used, to validate the myosin modulation in our ventricular myocyte model. And you can see, most of you know omecamtiv mecarbil. So this is a compound to treat HFrEF in phase 3 clinical trial. Mavacamten has been recently approved by the FDA for the treatment of obstructive hypertrophic cardiomyopathy. And also, I'm listed other compounds that they are research tool when it comes, to inhibit the myosin. Here is some data, showing the ability when you inhibit the myosin to decrease contractility. And you can see here we tested blebistatin, 3 different analogs of blebistatin, and also we tested BTS. So all these compounds are known to inhibit the myosin. And what you can see here, you can see blebistatin and BTS, for example, they cause concentration-dependent decrease in contractility. And you can see also that we have good reproducibility of data when we tested, for example, BTS multiple times, from the same isolation of a myocyte. But what you can also see with the analogs of blebistatin that, they behave differently compared to blebistatin, and this is what will give you SAR data to support to support developing better your chemistry. And this is how mavacamten will decrease contractility and treat hypertrophic cardiomyopathy. So basically what you see is that during hypertrophic cardiomyopathy, you have a lot of myosin head, that they induce hypercontractility. And when you block the myosin with mavacamten, basically you will reduce and control better this hypercontractility. And on the right-hand side of the slide, you can see the data we obtained, assessing the effect of mavacamten in our ventricular myocyte. You can see we tested this in 2 different donors. We can see a nice dose-dependent effect, and most importantly here, that our models predict, the clinical effect of mavacamten, but also it predicts the dosing of mavacamten in the clinic. The dosing of mavacamten in clinic is around 200, 300 nanomole, which correlate well with the IC50 that we obtained in our experiments. This is basically how omecamtiv works. Omecamtiv, in contrast, or mavacamten is an activator of the cardiac myosin. And this slides gives you an idea how omecamtiv works basically. So omecamtiv will speed up the ATP hydrolysis and will binds with high affinity to the myosin heads containing ADP. This will stabilize the myosin head, and this this will lead to a greater force in each cardiomyocyte contraction. And this is basically our data with omecamtiv. You can see when we did the dose response curve, we can detect the effect of omecamtiv in increasing contractility. We can also predict the dosing, the therapeutic dosing. And you can see also we tested the compound omecamtiv in multiple donors. You can see we have a good reproducibility of data. So I will compare omecamtiv to effect of calcium on contractility. Of course, when you increase the calcium by treating the cells with extracellular calcium, basically you will increase contractility like you increase it with omecamtiv. But the two compound really, or the two treatment really behave differently. You can see when it comes to omecamtiv, looking at the effect of omecamtiv on different parameters that you can derive from the contractility transient, you can see that omecamtiv will, for example, increase the time of the contraction in addition to increasing the contraction itself. When you look at the effect of calcium, you can see that the calcium treatment, despite that it increase the contractility like omecamtiv, it has no ability to modify or modulate the parameter of the contractility transient. So this is how you can start deriving some mechanistic information when you testing different drugs and try to better understand how they are behaving basically. Again, what is known about the myosin inhibitors that they do inhibit contractility without affecting intracellular calcium. And this is a good example with mavacamten. MYK was the research name for mavacamten. And you can see at the therapeutic exposure, the compound did not show any ability or potential to modulate intracellular calcium. Verapamil as a calcium inhibitor, you can see we were able to show that verapamil was able to decrease intracellular calcium and therefore decreasing contractility. And this is the summary data when we looked at all these myosin modulators. You can see none of them showed any ability to modulate intracellular calcium, where our positive control ISO as a beta-adrenergic agonist and verapamil as a calcium antagonist, both of them showed ability to increase and decrease contractility. This is a way, to show that the cells were responding to positive controls. And again, then after, as I said, we can procure diseased human heart. So this is where we looked at the effect of omecamtiv on HFpEF and HFrEF. You can see the difference between ability of omecamtiv to increase contractility in HFrEF condition versus HFpEF condition. And this explains the reason why omecamtiv is being in phase 3 clinical trial to treat heart failure with reduced ejection fraction. And you can see again, when you look at its ability to increase the contraction time, you can see omecamtiv works better in HFrEF condition compared to HFpEF condition. And again, this is the kind of data that we can provide to your programs to try to support a better translation when your compound will be tested in clinical trials. Again, this is another piece of data with mavacamten, and again here we did the same. We did compare the effect of mavacamten to decrease contractility in addition to normal hearts in HFpEF and HFrEF, and you can see clearly that mavacamten can control or decrease contractility better in a HFpEF condition versus HFrEF condition. And this is not a surprise that BMS and myocardia now they have a phase 2 clinical trial to assess, the potential of mavacamten to treat HFpEF patient. Remember here, what you need to do with HFpEF patients, so you need to control better, the activity of their hearts, and also mavacamten. Based on the data I will show, it will help them, control any arrhythmias that may lead to cardiac death. I will show you some data to tell you, that if we use contractility is a good approach, to go after compound to treat arrhythmia. for many years now, people used only electrophysiology and going after effective refractory period, basically to try to demonstrate the ability and the potential of a drug to be anti-arrhythmic. What we published here, and the data that I will show you today, that contractility also can be used as a model, to go after anti-arrhythmic, novel anti-arrhythmic drugs. So this is some data we published in scientific reports in Nature. So basically what we did here, we validated the idea that late sodium current inhibitors, can be used as anti-arrhythmic drugs. And here what we used ranolazine. As you know, ranolazine is in phase 2 clinical trial for treatment of atrial fibrillation, but also we did use another compound from GS967 from Gilead Therapeutics that was being developed as a next generation of ranolazine. And you can see here what we did. Basically, we did record contractility and normal myocyte. And then we did use ATX, which is a toxin to activate late sodium current. So what you see here in figure B, we do induce arrhythmia. When we bring the GS967 to the game, basically we block the arrhythmia that was caused by ATX. And by the way, ATX is a tool compound to mimic long QT syndrome type 3 arrhythmia. And you can see also we made the same observation when we tested ranolazine and showed that ranolazine was able also to block the arrhythmia induced by ATX2. After this, we moving from long QT3 syndrome, we moved to long QT type 2 syndrome arrhythmia. So this is where you block the Hurge channel, or you block the IKR potassium current in the human heart. And again, you see here when we used a selective blocker of the Hurge channel, we were able, to induce after contraction, which you see them in figure B. And again, when we combine E4031, the selective blocker of the Hurge channel, with the GS967, you see we were able, to block completely the incidence of arrhythmia in this myocyte. So this data shows you that we can modulate and block ventricular arrhythmia, but also from the human heart, in addition to isolate ventricular myocyte, we can also isolate atrial myocyte, and this is where we can go after atrial arrhythmia. Again, we repeated the same experimental design here. We did induce arrhythmia with ATX2 to mimic long QT syndrome type 3 arrhythmia. And again, when we treated the cells with GS967, we were able to completely block arrhythmia in this atrial myocyte. So all this data comes from ventricular atrial myocytes that we isolated from normal hearts. But again, we can procure donor hearts or affected by atrial fibrillation. What you see in figures A and C. So this is a baseline recording of contractility, and we were able, to record arrhythmic markers and event. And you can see one of the markers in figure C is a premature atrial contraction. So this is what was presented this morning as a PVC at the level of ventricles. And you can see, so this arrhythmia at baseline recording are a testament and demonstrations that the atrial remodeling did happen in this patient. So there is no need here to induce the arrhythmia. And you see when we did treat the tissues with 967, we were able to block arrhythmia and to restore the atrial function to its normal activity. So now I will move a little bit to the myosin inhibition. So we did something similar. We did observe, that myosin, has some potential to block arrhythmia. So we wanted to go ahead and investigate this in more details. You see here again, we used ATX2 to induce long QT type 3 syndrome arrhythmia. Again, you can see in figure B, we were able to induce the arrhythmia. And when we used mavacamten here at 0.3 micromole concentration therapeutic exposure, we were able, to block completely the arrhythmia. The next type of, so this is the summary data with ATX2. We did use, as I said, mavacamten at therapeutic exposure and also at the threefold therapeutic exposure at 1 micromole. And you can see, we were able to reverse or block completely the arrhythmia in this experiment. Also, we did demonstrate the ability of blebistatin also to block arrhythmia in the same experimental condition. Next, we also induced arrhythmia by long QT type 2 syndrome arrhythmia using, again, dofetilide as a selective blocker of the hERG IKR channel current. And again, you can see we were able to induce the after contraction, our arrhythmic event here, and we were able to block the arrhythmia with the mavacamten at 1 micromole. Also, we did we tested multiple different scenarios for arrhythmia, and here, we did test the scenarios of decreasing relaxation reserve arrhythmia. So I will try to explain a little bit what is the relaxation reserve. So this is the action potential repolarization reserve, and this is maintained by the hERG channel and by KV-LQT potassium current. So the reserve is there to protect the heart against any triggered arrhythmic activity. So to make this reserve weak, basically, and to mimic what happened in patients, we did block the hERG channel with dofetilide, and also we did block KV-LQT1 channel with HMR 1556. And you can see when we used the combination of the two blockers, we were able to cause or to induce alternance. And again, alternance is a very well known arrhythmic markers when it comes to human heart arrhythmia. And you can see, the data when we look at the contractility transient. We have a huge beat-to-beat variability, if you want, in the contraction. You have strong contraction followed by small contraction. So this will create abnormal ventricular rhythm. And you can see, when we tested mavacamten in the presence of these two blockers, we were able to completely inhibit the alternance and recover a normal ventricular cardiomyocyte function. Again, we tested this also in a HFpEF donor hearts. Again, you can see here we induced arrhythmia with ATX2, and when we tested mavacamten in the presence of ATX2, we were able to completely block the ability of ATX2 to induce arrhythmia. So this is another piece of data with mavacamten and the myosin inhibitors. As you can see from the title of the slide, that myosin inhibition reduces sympathetic stimulation. So if you have a high drive of beta adrenergic stimulation, basically this is a potent stimulus for generation of arrhythmia. So what you see here when we tested or mimicking the high adrenergic stimulation with ISO, you can see we increase contractility, and using mavacamten at therapeutic exposure, at 3-fold therapeutic exposure, we were able to significantly reduce the beta adrenergic effect on a human ventricular myocyte. And we can see the effect if you test ISO first and bring the inhibition, or if you inhibit the myosin first and then apply or bring in the adrenergic stimulation. But more importantly here, you can see that we were able to block the arrhythmia that was caused by the adrenergic stimulation. And an important message here, HFpEF patients have been treated for many years with beta blockers. And beta blockers for this patient is not a good treatment, so it doesn't really treat HFpEF well. In addition, that beta blockers, they are associated with a lot of side effects. So this data will have an impact on how mavacamten will be used to treat patients. So in this case, if you use mavacamten, because of its ability to reduce beta adrenergic stimulation, there is no need to use beta blockers when you treat this patient. So you will help them a lot by experiencing less side effects. So this is a summary of the data that arrhythmia can originate in the ventricles and the atria. And again, conventional anti-arrhythmic drugs often lack efficacy, but also are associated with many side effects, and one of the side effects is proarrhythmia. So these drugs that target ion channels, they were developed to stop arrhythmia, but at the same time, their side effect, they will induce proarrhythmia and arrhythmia again. And our data indicates that the inhibition of the cardiac myosin can be considered as an innovative approach to anti-arrhythmic drug therapy. And if this data will be confirmed in a clinical trial, this really will be a potential game changer when it comes for the treatment of cardiac arrhythmias. And to close, the presentation, this is a take-home message from me and from AnaBios. Ex vivo human cardiac platforms are now available. They can be used, to study drug effect in human heart cells and tissue. And certainly, the data being generated at preclinical stage will allow you, to make the right decision if you want to take your compound to clinical trial, or if you want to stop your compound and try to get a better compound with better qualities. So please use them. They will have a tremendous impact on your clinical program and your ability to launch a novel treatment to the market. Okay, thank you.